So now on with the early careers talks. First up, we have Mark Woolam. He's from the Integrated Nanosystems Development Institute. He's also a researcher at Indiana University, Indianapolis, where he's focusing on developing methods to sample and identify volatile organic compound biomarkers in exhaled breath. Mark completed his PhD, where he concentrated on utilizing chemometric approaches to identify VOC biomarkers both in urine and in breath for diseases including prostate cancer, breast cancer, hypoglycemia, COVID, and cystic fibrosis. Upon graduation, he joined the INRI, where he continued to drive scientific discovery, demonstrated through publishing articles in high-impact factor journals and helping to secure several extramurally funded awards. So Mark's talk title today is Establishing Healthy Baselines to Harness the Potential for Breath-Based Biomarkers for Non-Invasive Health Monitoring in Diverse Medical Applications.
Hello, all. My name is Mark Woolam. I'm a senior research scientist here at Integrated Nanosystems Development Institute at Indiana University, Indianapolis. And today I'm going to be talking to you all about establishing healthy baselines to harness the potential of breath-based biomarkers for non-invasive health monitoring applications. So a little bit about the analytical methods used for VOC detection and exhaled breath. The gold standard is gas chromatography mass spectrometry, or GCMS, because it has high capability of chromatographically separating, structurally elucidating, and quantifying volatile analytes in complex sample matrices. However, this technology is not ideal for point-of-care detection because it occupies a large footprint, it's expensive, and it requires trained personnel to run assays. Therefore, for end application, portable nanosensor arrays integrated with machine learning algorithms are the best case scenario. Therefore, our lab's unique approach is to use the information from GCMS regarding biomarker discovery to inform the development of miniaturized gas sensors that can detect the biomarkers with not only high sensitivity, but also high selectivity. So for GCMS analysis, we need a method to capture or trap the VOCs from breath. And up until this point, we primarily use solid phase microextraction, or SPME, using two different methods. The first method is called the cryothermal transfer. And here we collect breath into a Tedlar bag, and we cryothermally transfer the VOCs from the bag to a headspace vial loaded with glass wool. And we do this using a mass flow controller with a vacuum. Once the VOCs are captured in the headspace vial, we can run our typical SPME GCMS assay. We also do Tedlar SPME, which is another method where we simply collect breath into the Tedlar bag, and then we expose the SPME fiber directly in the bag, and we can capture VOCs from breath this way as well. So using the cryotransfer process with SPME, we have profiled VOCs and exhaled breath for many different medical conditions. Just for an example, you see hypoglycemia results in the top of this screen. We identified a biomarker panel of just 6 VOCs that could distinguish hypoglycemic events with over 90% accuracy, and we blindly verified this result in hundreds of samples. For COVID-19, this study, we had a much smaller patient cohort, but nonetheless, we identified many volatile analytes that were significantly enriched in COVID patients. And we identified different panels or biosignatures of VOCs that could distinguish all COVID patients with very high accuracy. There was preliminary evidence to suggest VOCs in breath could distinguish COVID patients based on severity or the number of symptoms. However, the coolest result from this study came after collecting and analyzing the breath samples from COVID patients 2 months after they recovered from the disease. And as you can see in the lower right-hand corner, the COVID recovered patients are really clustering with the healthy controls, demonstrating that VOC biomarkers of COVID-19 in the acute phase are restored to healthy baseline levels upon recovery. And these results were really the motivation for the current project that I will be talking to you about, which is establishing healthy baselines of exhaled VOCs. And this project was funded by the Defense Threat Reduction Agency. So for this project, we actually developed a new method to sample VOCs from breath called direct breath SPME or DV-SPME, where a volunteer exhales through a viral filter down onto an exposed SPME fiber that can absorb the VOCs in breath. We published this paper in Molecules, and we quantitatively compared it to the other SPME-based approaches that I previously talked about, Tedlar SPME and cryotransfer. The results showed that across the board, cryotransfer was the most sensitive method. However, if you're focusing analysis on some smaller VOCs like acetone and isoprene, DB-SPME or Tedlar SPME may be better situated. Nonetheless, using this method, we collected and analyzed 164 different breath samples from unique individuals. We analyzed their breath samples using a portable or handheld nanosensor array. We also performed a longitudinal study where 10 of the cross-sectional volunteers provided an additional 9 samples over the course of 6 months. And in addition to the breath analyzed by GCMS and portable sensors, we also collected blood samples for comprehensive metabolic panels and complete blood counts. And we also collected other data on confounding variables using a lifestyle questionnaire. So first, I'm going to be talking about the GCMS results. And after instrumentally analyzing all the samples, we spectrally aligned the GCMS chromatograms and identified over 2,000 VOCs. However, we had to remove duplicates, VOCs that were coming from the SPME fiber and or other collection materials. And we also removed VOCs with low abundance / frequency. This led to the identification of 156 VOCs. Then we created 3 different filters. VOCs that were at least in 10% of breath samples with greater intensity relative to the background. Then we identified 32 of these VOCs to be statistically significantly enriched in breath samples relative to background. And then the final filter where we have 17 of these compounds that were present in at least 50% of the breath samples. So after we identified these VOCs, first using the filter with the greatest amount of compounds, the 62 VOCs, we performed functional group frequency analysis. And overall, we saw many aromatics, terpenes / terpenoids, as well as carbonyls. For further elucidation, you can see the chemical structure of the 17 VOCs in the upper right-hand corner, and these are the compounds that were present in at least 50% of breath samples. After performing the functional group frequency analysis, we wanted to visualize the expression patterns of the core set of 32 VOCs, which were significantly enriched in breath. So in the hierarchical heat map in the upper left-hand corner, you can see the VOCs are listed as rows, the samples are listed as columns, and not only could you see the relative intensity of each of the VOCs, but you can also see how they cluster based on similarities in expression among the data. Very interestingly, all of the monoterpenes clustered together, and you can see this in the yellow highlighted portion. We have added correlation plots showing significant correlations between alpha and beta pinene, isoprene and alpha pinene, and there are many others. Below, there is a correlation matrix showing and highlighting the significant correlations among the terpenes. And overall, we think that the clustering may be due to similar biological origin. Next, we sought to benchmark VOC reproducibility in the cross-sectional cohort or inter-subject variability, as well as the longitudinal cohort or intra-subject variability. On the upper left-hand corner, you can see in both cohorts, VOCs are definitely variable. However, cross-sectional measurements were significantly more variable than longitudinal. Nonetheless, there was a correlation between the two. VOCs that were more variable between people were also more variable within one person over time. So both of these results kind of brought up the question, do individuals have a characteristic VOC baseline in breath? So to answer this question, we did principal component analysis on different filters of VOCs. And in all the analyses, the replicates of different volunteers could not be distinguished. So no, individuals do not have a characteristic baseline. And this is further mitigated by the fact that when we compared the longitudinal to the cross-sectional, they all overlapped. So why is this? Do we have a discrepancy? Why are VOC measurements more reproducible within a single person, but not a single person has a unique characteristic baseline? To explore this, we looked at the signals of individual VOCs, and you can see this in the lower right-hand corner. In all of the VOCs, the cross-sectional range is greater than any single volunteer. However, they all overlap to a high degree. So that pretty much, reconciles what we just saw. Finally, the last step that we took regarding the GCMS healthy breath dataset was to analyze it for VOC correlations with confounding factors of interest, i.e., sex, BMI, age, etc. Across the board, for all of the biostatistical comparisons, we saw limited to no correlations with confounding factors after we accounted for false discovery. The exception here was biological sex, where we saw isoprene was significantly elevated in males relative to females. However, when you analyze all of the VOCs in the breath print using unsupervised multivariate analysis, you cannot differentiate males and females to any degree. Taken as a whole, this really shows that confounding variables that are not related to disease diagnosis do not have an impact on healthy breath VOCs, at least in this cohort. And it should be noted that this cohort was relatively young and did not display heterogeneity in many confounding factors. In addition to the GCMS analysis of healthy breath VOCs, we also used a portable nanosensor array composed of 8 metal oxide sensors. From each of these sensors, you can extract a total of 17 features that correspond to the detection of different exhaled VOCs. You can see an exemplary heat map of these features in the upper left-hand corner, where the samples are listed as columns. When you look at the reproducibility of these features in the intersubject and intrasubject datasets, or cross-sectional and longitudinal, again, it becomes very clear that cross-sectional measurements are significantly more variable than the longitudinal data. Furthermore, when you use the nanosensor data to try to distinguish different volunteers, you can clearly see the data overlaps. And this really mirrors the results from the GCMS Healthy Breath study. Once we benchmark nanosensor feature reproducibility within the Healthy Breath dataset, we next wanted to correlate the features to specific VOCs that we detected and quantified by GCMS to really understand which chemicals the sensors are responding to. So to do this, we undertook machine learning in both datasets, and we also undertook penalized canonical correlation. You can see the results for the first 10 canonicals below, which correspond to multiple sensor features and multiple VOCs, and they all displayed higher statistical significance and degrees of correlation relative to analyzing VOCs and sensor features alone. And the results of these multivariate analyses really indicate that cyclohexane and isoprene are the main players and the main contributors to the observed sensor signal. The last thing that I want to mention about this study is, in addition to healthy breath samples, we also collected healthy urine samples from the same cohort, and we are currently analyzing this data in a similar fashion to the results that you saw for healthy breath today. However, we did quantify acetone in the urine samples, and we correlated it to exhaled acetone levels. And here you see a very statistically significant correlation between the two, which is very exciting. Before I wrap up today, I just want to talk about some of the future research projects that are going on in our lab. First, we were just funded by the American Cancer Society to translate portable GCMS systems to enhance prostate cancer screening. We have previously published results on VOC biomarkers that we identified using a benchtop GCMS system, and this project really aims to translate and optimize portable GCMS systems to detect these same signals. In addition, we were also just funded through the NHLBI to identify breath-based biomarkers for pulmonary exacerbations in kids with cystic fibrosis. Not only that, but we are also trying to see if certain signals in breath could determine if patients are going to need antibiotics or respond effectively to antibiotics. Finally, I would like to acknowledge the amazing team that I have behind me. My main mentor, Dr. Mangalal Agarwal, is an amazing leader to everyone in our lab. And specifically among the grad students, I would like to thank Arai Scholes and Shivam Haranjal, who did a lot of heavy lifting in the Healthy Breath study. I would also like to thank our collaborators on the Healthy Breath study, who are Doctors Ryan Relic, Shah Kao, and Shia Owen Liu. And finally, I would like to also acknowledge our funding sources, which are the Defense Threat Reduction Agency, the American Cancer Society, the NSF, and the NIH. I'd also like to thank Alstone Medical for hosting this amazing conference. And I'd like to thank all of you for taking the time out of your day and attending. I look forward to the rest of the conference, and I hope you have a great day.
Thank you, Mark, for an excellent talk there. And thank you also for joining very early your time. I know there's a lot going on in the US at the moment, so hopefully you managed to get some sleep.
I'm always here in the lab this early.
Great to have you here. Yeah, so really nice talk. I was impressed actually with your COVID data. I hadn't seen that myself, where you saw a reversion back to a more healthy level at the end of the illness. So I just wondered, did you identify any of the constituent VOCs there as part of that panel, and did it line up with what had been reported at all by other groups working on COVID?
Yes. So I know that we identified this aldehyde 355 trimethylhexanal. So I don't know if anyone particularly identified that specific molecule, but I know aldehydes in general have been. We also identified a terpene, a sesquiterpene, cedrine, and an ester. I'm blanking on but it's in the paper. And we did run peer standards of those compounds for verification as well.
Great, thank you. Another question here. I know you've done both the urine profiling and the breath sampling within your healthy study there. Have you had a chance to see whether there's any concordance between the two at all yet?
So that's something that we're really digging into right now. We're really excited about the acetone correlation because that kind of helps validate both of our findings, I feel like, on both sets. And it's just interesting that you can look at either place and end up with the same answer. Overall, though, we are seeing differences in the profiles. The main takeaway I see so far is there's a lot of terpenoids in urine and terpenes in breath. And we're really trying to draw correlations between those two. There's a lot of data that we're just trying to flush out.
Yeah, thank you. And also, I might have missed it, but did you take a food diary as part of your healthy control study? And were you seeing any correlations between food intake and volatiles?
So this brings up a really good question. And maybe like this will open up the floor. Because before we started, we were like, okay, we want to track food intake 2 days prior to breath collection and see how this can impact the breath profile. But once we got the data, we really didn't know how to handle it and make correlations to certain foods and the VOC outputs. What we did is we correlated different diets, like general diets, and we didn't see any large correlations there. But yeah, I mean, I feel like that's something difficult, and that's something we struggled with, and maybe as a community we can come up with a better way to quantitatively track the impact of food and diet.
Thank you. Another question here. Did you look at smoking at all? Were any of these individual smokers? I'm guessing that would give quite a strong profile.
Yes. So there was a small percent of smokers. I think there was like 10%, around 10%, maybe less. And we looked for significant differences, which is difficult because we have a small number of samples, uneven distribution of samples. But regardless, after reconsidering, we really needed to ask more questions about smoking. And we did actually go back and ask more questions about if they were smoking cigarettes, using vaporizers, tobacco, marijuana. And they were just so heterogeneous. So the smoking group was so small, and then the smoking group was so heterogeneous that it was hard to track. the impact is, and the majority of them were users of vaporizers, which I feel like can be, depending on the flavor, uh,
Mm,
impact the breath profile a little different. So
for
I think
sure.
we need more dedicated studies, like focus studies on different flavor profiles of e-cigarettes, which I think a lot of researchers are already doing.
Absolutely.
But
And then also looking at the health effects as well. Excellent. And then in terms of the volatiles you were measuring, do you get a sense of how many of them were truly endogenous versus introduced exogenously?
yeah, I'm gonna take Alstone's amazing lead on there. I'm gonna keep using on breath, and I think I think we could say from this study, we using this method, the DB Spearman method's not that sensitive. I'll tell you that right now. We detected 32 VOCs on breath, um, and I'm not going to speculate if they're endogenous or exogenous. I think that in the clustering of the terpenes, I said biological origin. I meant to say origin in general. We're speculating it could potentially be both. We think terpene production could be endogenous and exogenous, but we don't know.
Yeah, perfect. Okay, I think that's it for now, Mark. Thanks again for joining us, for answering all the questions and getting up early. Really appreciate it. And see you soon.
Thank you.